Let's move along. Our lunchtime is getting shorter, and I do want to give all the speakers time for their presentations. Our next speaker is Simon Egley. He is a mycorrhizal researcher from the Swiss Institute for Forest, Snow, and Landscape. And he has done a lot of work with long-term studies of mushroom harvesting, mushroom diversity, the impacts of harvesting. And more recently, he's done some research on thinning of forests and the effect on productivity, and also the effects of climate change. Let's welcome Simon.
Thank you, Dave, and I want to thank also the organizers for the invitation. It's a great pleasure for me to be here and to present you results on mushroom production and diversity under experimental thinning and climate change. So I will talk about two factors which influence mushroom production, mushroom yield, thinning and meteorology. The data on which I base my presentation come from a fungus reserve in Switzerland. The reserve was established in 1975 to allow microecological research, field research. It's 74 hectares, and it's in a typical representative forest for the Swiss plateau. So it's a common forest type. It's a mixed beech forest with beech as a dominant species. The fruit bodies, all the fruit bodies were inventorized from 1975 up to 2006. It's a full inventory of all the Epigeas macromycetes. And the relevés were done weekly from beginning of the season, from May until the end of the season, December, until the first snowfall or frost. The spatial resolution is 1 square meter, so it's like a mapping of the fruit bodies. And then additionally, the fruit bodies were marked with a blue point, with a blue color, to avoid double counting in the following week. And the plots were enclosed by fences to prevent them from mushroom pickers and from animals, big animals. A total of more than 100,000 fruit bodies were monitored in these 32 years, belonging to 450 species, 273 mycorrhizal species and 177 non-mycorrhizal species. One of the experiments which I want to present you is a thinning experiment in 1987. So a very dense old growth plot was thinned. You see here the picture before and after the thinning. So all the big trees were cut out. The trees of the canopy were cut out, and some single trees were left. And all the understory was left. Here the situation before thinning and after thinning. Before thinning, the stand was dominated by these large trees, these big, very old, large trees. and Fagus sylvatica in the understory. Then after the thinning, the large trees were cut, nearly all, and the Fagus sylvatica was the dominant species, so the understory got dominant. And total basal area was reduced from 124 to 64 square meters. That's quite high basal area. Here are the results of the thinning. The mushroom production was very low in the period before thinning, and it increased sharply after the thinning, but with a delay of about 3 years. 4 times more species and 10 times more fruit bodies in the mean, annual mean, over these 2 periods. And the mycorrhizal species, here the blue bars, they reacted more pronounced even. So they developed 5 times more species and 17 times more fruit bodies. And because we are interested in the relationship between tree growth and mushroom production, we took tree core samples, so to measure the tree rings as a measure for the growth of the trees. And you see here the 2 lines. The green line represents the beech tree rings, so it's the mean value of all the beech trees on the plot. So you see an increase in growth just after the thinning. And these trees were suppressed by these old trees, which were cut off, and they were exposed to the sun, and they began to grow immediately after thinning. Instead, the large trees, they continued their low growth. The big, large trees left on the plot. And to know more about the relationship, the causal relationship between these 2 reactions, we looked more in detail to the community of the fungi, of the mycorrhizal fungi. And we found really the answer in the comparison of the community before and after the thinning. Before thinning, the strictly beech-specific mycorrhizal fungi, they had a proportion of 19%, and they increased to 70% after the thinning. So the strictly beech-specific fungi, they increased in fruit body production, very dramatic. And in other plots, in control plots, no such development was observed. So this is an example where we can really see a relation between tree growth and production of mycorrhizal species, associated species. In the literature, there are other examples, very nice examples, which show this relationship between tree growth and mushroom production. One is from José Antonio Bonet and his colleagues in the Pyrenees. They showed the importance of the basal area. So stands with a basal area around 20 square meters per hectare produce the maximum of fruit bodies. Then the results of Ortega Martinez and others, with data from Fernando Martinez, they looked at the age, the relation between age of the trees and production, and they found a relation. For Boletus edulis, they found the highest production in mid-age class stands, so between 30 and 70 years old. But for Lactarius, it's a different pattern. So Lactarius has the minimum production in this age class. Then some results on effects of thinning. An experiment of Luoma and others in Oregon, they showed that thinning in a Douglas fir forest caused a clear decline in fruit body production, but with great variation according to the pattern and the level of the thinning. So they had grouped thinnings and dispersed thinnings in different levels. So 100%, 75%, 40%, and 15% tree retention. Another study in a Pinus contorta forest shows that, so they looked 10 years after thinning and 17 years after a clear cut. They made an inventory of the fruit bodies. And they noticed that even 10 years after thinning and 17 years after clear cut, the species diversity was still reduced. The effects on fruit body production were very species specific. So they had species which reacted positive, species which reacted negative to this treatment. Then they noticed also that clear cutting reduced the fruit body production of the ectomycorrhizal species, even 17 years after clear cut, except one species, Suillus brevipes, which increased its fruit body production. So it seems that this species found good competitive conditions in this treatment. Then another study in a hemlock red cedar forest, no clear effects on species richness and productivity. So they found positive, neutral, and negative effects 5, 6, and 7 years after the thinning. An experiment in Great Britain. So they looked 12 years after a 50% thinning in a Scots pine forest, and only 3 species out of 19 responded to this treatment: Suillus bovinus, Confidius, roseus, and Cortinarius semisanguineus. And all the other 16 species showed no reaction. Then a study of David Pilz in Oregon. They looked to the chanterelle production after a thinning in a 50-year-old Douglas fir stand, and they found a significant decrease in the first year after the thinning. So a decrease in number and in weight. But they noticed a tendency of recovery. So this effect was very clear in the first year only, significant. Then a known result in a Norway spruce stand with plots with different stem densities. And in comparing these different treatments, we found that the medium dense Norway spruce plantation produced most of the fruit bodies, much more than the dense stand, and much more than the not dense stand. Okay, we could add here the results we heard before from Juan Martinez de Aragon, but they do not change the conclusions. The conclusions out of these thinning experiments, the available results, they are inconsistent. So we have positive, negative, neutral reactions, and depending on the structure of the forest, on the age of the forest, on many factors. So there is no clear tendency of reaction. Very diverse. And diverse species specific, so each species has his own reaction. But it seems that medium dense forests are most favorable for mushroom growth. Here there are no contradictory results. And we can conclude that thinning treatments alone do not fully explain the mushroom reactions. So there must be other factors involved. And of course, the meteorological factors are known to be very important for mushroom growth. So we looked to our data, we compared we correlated our 32 years data set with various meteorological factors, with rainfall, with temperature, with cloud cover, with drought index, and we tested also time lags, but without results. The only results we found are presented here. The only significant results. So for the meteorological data we used gridded high-resolution climate data which are available online for free. They're the data from the European Climate Assessment Dataset. And we made spatial field correlation with these meteorological factors. And we found, so perhaps to explain, these are the maps of Europe, and where there are colors, there are significant spatial correlations. And blue is negative correlations, and red positive correlations. And where we have black color, there is no significant correlation. So in the first map on the left, we see the June-July fruit bodies, so the early fruiting species, and they correlate significantly with the June precipitation, so the total of the June precipitation. The white star represents the location of the reserve. Then, for the August-September fruit bodies, we found a good correlation with the August precipitation. And for the late fruit bodies, October-November fruiting, we found a correlation with September precipitation. So fruit body production is driven by pre-rainfall of the current year. Then we looked to phenology. And we looked, it's on the right. We determined the weighted week number of fruit body appearance. And that's not the week with the highest fruit body production, it's weighted all over the year, And we found a very high correlation, significant correlation. So as higher the temperature is in August, as later the fruit bodies appear. So a clear phenological relation between August temperature and the fruit body season. Then meteorological factors can also have long-term effects. This is a paper of Genji and others. They analyzed herbarium data from Great Britain over the last 50 years, and they found that the mushroom season has expanded in both directions. So the mushroom season starts earlier and ends later, and it gives an expanded season, a longer season. And this can be observed since the 80s. And then they also noticed that some species began to fruit twice in a year. species which fruit normally only once, they began to fruit twice, so more fruit body production. So a positive effect of climate change, we can say. So of course they related this development to changing temperatures and changing climate. Then 1 year later, the Norwegian, they made the same. They looked to their data, herbarium data, but they found another pattern. So in Norwegian, the mushroom season begins to start later, and the tendency of the ending is not clear. Then we had a look to our data from the fungus reserve, and we found again another pattern. So 3 different patterns. And in our data show that the mushroom season starts later and ends later. So it's a shift forward. And here are the data in detail for all the species, for the mycorrhizal species and for the non-mycorrhizal species. And we see this tendency. Starting later, ending later. And if we put the most frequent, 25 most frequent species from our data in a graph with 4 quarters according to the slope of the first and the last observation, then these 25 species are located like that, these blue points. So the most of them are in this plus quarter, so later starting, later ending. And if we look to the data from Great Britain, they are in another coin of another quarter of this graph. And the Norwegian species, they are again in another. So 3 different patterns. And now we are interested in looking at other data sets from Europe, long-term data sets. And we plan to integrate the Spanish data from Fernando Martinez and from Finland, another data from Finland. Because we want to know what are the drivers for these different patterns over Europe. So the conclusions from these observations: good and poor mushroom years seem to be driven mainly by meteorological factors. Productivity by pre-rainfall and phenology by summer temperature. And in Switzerland, the mean mushroom fruiting dates has delayed by 10 days between 1975 and 2006. And the fruiting period remains unchanged, so first and last fruiting days have shifted forward. And these data show that the mushrooms seem to be sensitive indicators for climate variation. Thank you very much for your attention.
Questions?
Merci Simon pour cette présentation. Une question sur la partie concernant les éclaircies, la sylviculture. Il y a un paramètre qui n'a pas du tout été évoqué dans toutes les études que tu présentes, c'est la technique d'exploitation en elle-même. Je veux dire par là que selon l'organisation d'une exploitation, donc la circulation des engins, le type d'engin utilisé, la date d'exploitation, le type de sol, on peut avoir des effets très différents sur le sol, on peut avoir des effets très négatifs avec de la destruction de l'humus, du tassement des horizons superficiels. Et j'imagine que ça peut avoir un impact très fort sur la production de champignons. Donc, dans toutes les études que tu as présentées, est-ce que ces paramètres sont pris en compte ou jamais?
Non, je ne pense pas. Ils ne sont pas pris en compte. Mais quand même, il y a... Ça, c'est sûr que la technique de silvicole a une influence sur la flore fongique. Alors nous pensons que nous avons un délai de 2 à 3 années et nous pensons vraiment que c'est la disturbance, on a disturbé le sol et après ça commence à se développer, mais il y a un choc au début et ça dépend à la technique à la technique silvicole, je suis sûr. Peut-être aussi dans les données de David Pilz avec la chanterelle, je pense que cette réaction dans la première année, ça peut aussi être provoqué par la disturbance. But in this, there are no data about the technique in these papers. It's not described.
Let's take one more question from Jose.
Simón, en relación a los resultados publicados el año pasado sobre el experimento de claras en Suiza y en relación también a los datos presentados por Juan, uno de los aspectos que más llama la atención es que en vuestro caso, después de la clara, la respuesta sucede 3-4 años después. Mientras que en el caso de los datos presentados por Juan, la respuesta a la clara es prácticamente automática, ya que sucede un incremento o una variación de la producción prácticamente al mes. Mi pregunta es, ese aspecto que puede resultar sorprendente, el tiempo de reacción, ¿en qué manera crees que es debido a temas de carbono o sobre todo a temas de agua? Porque supongo que el agua en vuestra estación ahí en Suiza no es un limitante, mientras que seguramente en la zona que presentaba Juan es un limitante. Entonces, ¿de qué manera al eliminar árboles estamos, por tanto, favoreciendo una mayor disponibilidad de agua por parte de los árboles? ¿Cuál es tu opinión al respecto?
Yes, it's possible, first of all, that as Sebastian told before, that this delay is due to the disturbance. But on the other side, I don't know how they made their thinning. Perhaps with another technique, I don't know. But otherwise, I think that water is perhaps less a factor in Switzerland, in Spain surely, but it can be that the disruption of the carbohydrate flow to the roots needs some recreation, so they have to build up the reserves in the mycelium to produce again fruit bodies. I think it goes in this direction. So it's a carbohydrate problem. And perhaps it can depend on the age and the structure of the forest, of course, on the carbon reserves. So this can provoke different reactions or different time lags. I don't know.
Okay, let's move on and save the rest of the questions for the final. I would like